For example, it is a Disney map of with images of handwritten numbers, and images with the same numbers or clusters flow together and is also represented with colors. Here's the second form of map. It is a UMap embedding of 3 million words from Google News dataset. And is this, for example, a third form? It's a Disney map, which embeds different so cells of different types from mouse visual cortex. All of them are great visualizations since they reveal the relatedness between embedded objects. But they are actually missing this explanation part. So we would need to see what are these structures and groups in the cluster. So we can explain maps starting with known set of annotations. For example, here is the workflow where we select marker genes, and it triggers highlighting cells with express marker genes in the Disney plot. And changing this selection will propagate through the workflow, so we have thus enabling the marker gene-driven exploration. The second alternative approach is to identify clusters on the map and mark them with the enriched cell types. In the work presented here, we focus on explaining document maps. We use point-based visualizations of documents and annotate them with the cluster-specific keywords. There are several keyword extraction methods available. Some of the most popular are TF-IDF, which scores words based of documents related to the entire corpus. Yankee is one which uses statistical features such as spread of the word, position and the frequency, text rank and ray graph based method, and approach based on word embedding and document embeddings highlights words which has embedding similar to the embedding of the total of the entire document. In our paper, we compared those approaches in order to see which one performs better. We gathered 17,000 papers on the topic on the longevity from the PubMed, and 4,500 of them already had assigned keywords by authors. Those keywords are used as a golden standard for evaluation of methods. Here we see precision and recall graphs, and we assessed both scores for 1 to 12 best scoring methods for each document. The best performing method is Yake, but surprisingly, the second best performing method is TF-IDF. Despite its simplicity, it actually outperforms some more sophisticated graph-based and embedding-based methods. In our case study, we used almost 17,000 abstracts of the papers. We embedded them with fast text embedding and used Disney to get point-based visualization. And on this visualization, each dot is a document. And here on the image, we can already see that some groups are formed, and these are groups of the similar documents. But we don't know what these groups actually are. So in the second step, we compute clusters of this embedding. We used Gaussian mixture model, sorry. And we assigned each document to more likely Gaussian component. If the probability was below the threshold of 0.6, it was not assigned to any cluster. We could alternatively also use some other techniques like k-means, but k-means has issue with cluster shapes and also dbscan, but dbscan is very sensitive to parameter changes. And in the last step, we annotated those clusters with keywords. We found 20 best scoring keywords for each document. According to cluster frequency, we first scored them and then we selected them based on cluster enrich, if they are cluster enriched. So we computed p-value according to the hypergeometric test, which compares the frequency within cluster and outside the cluster, and it was lower than 0.5, the word was selected. We found our keywords meaningful. For example, if we take a look at the cluster number 5, we can see that here are general articles that study longevity, and the central titles according to the Silhouette score are population aging and public health, the active aging concept, who were old in the Middle Ages, and longevity according to the life histories of the oldest old. Differently, the cluster number 8 represent model studies on longevity. And according to our intuition, also other cluster labels equally well represent clusters topics, so topics of the documents, which supports the value of our approach to exploration. We are implementing this approach as a part of semantic text analysis tool, which is part of Orange. Orange is a toolbox for interactive explanatory data analysis. And we already have highlighting of documents according to the list of words. Currently, we are implementing the other phase, so the concept which is presented here in this paper, where we highlight clusters in the map and we mark it with keywords of this map. Similar concept was already used for annotation of single cell data, which you can see on the image. Development of presented method and tool and corresponding tool Orange is held by Bioinformatics Laboratory at University of Ljubljana. So I would like to thank my colleagues. And the work was supported by grant on text semantics analysis by Ministry of Public Administration in Slovenia. I also want to thank organizers for this workshop for the opportunity to present this work, and you audience for the attention. Thank you.